Thank you, Martin, for the introduction. So, hello, everyone. My name is Nicholas, and today I'm going to share with you some details about the production and the purification of recombinant antigens. So, we're going to see together some challenges and solutions. So, first of all, what's an antigen and what's a good antigen? So by definition, an antigen is any substance that will induce the immune system to produce an immune response against it. So as you can see on the figure on your right, you can see that antigens can be from various sources. A good vaccine antigen should be located at the cell surface or secreted by a pathogen, for example, like a toxin. A good vaccine antigen should be accessible for immune cell and obviously be highly immunogenic to be very effective. It has to be different, has a different structure from the host proteins, and such antigens are mainly proteins or polysaccharides. So I wanted to start with a quick overview of recombinant antigens production. So the first step is to identify your antigen of interest and to obtain its DNA sequence. So to obtain its gene, just kind of put so this is our first step. And then the second step is to clone the gene sequence into an expression vector and transform this vector into the expression organism of your choice. Step number 4 is to cultivate the host organism to a high cell density and then induce the protein production. And the final step is the isolation and the purification of your protein. So let's start with step 1, which is the identification of your protein of interest. This step can be achieved using the reverse vaccinology concept, which basically rests on the use of the genomic information to assess the complete antigen catalog of an organism without having to culture it. So basically, you will start with your pathogenic bacteria or viruses, and you will isolate the genomic DNA from the cell. And then, using bioinformatic analysis, you can identify your protein of interest. So once you have your gene, the second step is to clone this gene into an expression vector. So here you can encounter several different challenges. So first of all, there is the host codon preference and the sequence composition. So for the host codon preference, you have to keep in mind that it's not every host that will use the codon in the same way. So for that, you can, the first solution would be codon optimization, which consists of modifying your protein sequences in order to respect the host codon preference without modifying the amino acid composition. Other solution would be to remove your regions that are not exposed at the cell surface, or even remove highly hydrophobic regions, because this region can because these regions can be translated into a protein that is not stable and not soluble. The next step is the transformation step, where you would put your expression vectors into your host organism and select the transformant on agar plate with a selective marker, such as an antibiotic. Here again, you can face several challenges. So once the transformation is done, how will you choose the appropriate transformant? So there's the selection of transformant. There's also the selection of an appropriate expression host. And there's also one problem could also be that the protein that you chose is toxic to the host organism. So what I would recommend for this step is to really take your time and take a look at the different hosts and different strains that are available, because they all have different genomic feature, and you can choose the one that is more suitable for your needs. As for the antigen toxicity, if it's the case for your protein, you can choose a vector with highly regulated or even a weak promoter. So that way it will prevent the leaky expression of the toxic protein. Then you will be able to reach a high cell density before doing the protein induction. Now for the selection of transformant, it is highly recommended to choose at least 3 different transformants on your agar plate, because different transformants will express your protein of interest at different level. So you can choose at least 3 different and assess the protein expression efficacy and just select the best one for your needs and select it for the further experiments. Step 4 is the high cell density culture and the protein induction. So in this step is kind of critical because you have many challenges that you can face. So you have to keep in mind that different parameters such as growth conditions and medium composition will highly affect your protein expression and purification. So in a growth conditions, you have to acknowledge the temperature, the pH, the incubation time. And for the medium composition, you have to look at different nutrients that is available for your host. Also, you can look at the inducer concentration. So it has been shown that some hosts prefer higher or lower inducer concentration, and all of these parameters will affect the protein stability inside of your host and also the protein solubility. So for the solution, there is really no miracle recipe to express and easily purify a protein. So you will have to do some trials with different parameters and see for your protein and the host of your choice what are the best parameters to do the protein expression. In case where it's not working, you could also try to add cofactors to the medium to add some salts or nutrients that will enhance the production of your protein. So basically will help the bacteria to grow. It has been shown that lower temperature and lower inducer concentrations are good to improve the protein expression because it forces the host to produce the protein at a slowly rate, and then make sure that there's no mistake in a proper folding. Finally, you could also add what we call a solubility tag, or a fusion protein that is really soluble, and that will also enhance the protein solubility and be easier for you to purify it. Final step is isolation and purification. So again, there's many challenges that we can face during this step, such as inefficient cell lysis, insoluble proteins, contaminants, and poor purification. Sorry. So if the method of lysis that you chose is not working, you could always try to use a different one. So there's many different mechanical and chemical lysis technique that could be more suitable for you. If you are really stuck with what we call inclusion bodies, and you don't have any budget to go back and modify your protein sequences, for example, you could use a solubilization buffer, which basically consists of denaturing the protein aggregates in order to purify them. But you have to keep in mind that these steps requires another refolding step, because you have to put the protein into its native structure. You could remove contaminants using the gel filtration technique or dialysis to remove, for example, imidazole salts. And to overcome the poor purification, you could add an affinity tag. So this is the famous 6X histidine tag that's working with the affinity chromatography. And this is a really good technique to purify your protein of interest. And if this is still not working, there's also many others chromatography types, such as ion exchange, for example. So as conclusion, we saw that the antigen production steps seems pretty easy and pretty straightforward when you look at it. but there's a lot of surprises that may occur at each step of your production. There's many factors that can be modified or added to enhance the production of a stable and soluble protein. But you have to remember that each protein is unique and has its own production whims. And finally, the production of a protein needs optimization, and it takes several trials and a lot of determination. But the important thing is to not give up and keep trying until you can purify your protein of interest. So with that, I'm sharing with you some references, and I would like to thank you all for your attention, and I'll be happy to answer your question.
Thank you very much, Nicolas, for this excellent overview of regarding protein production and its many challenges. I have a question regarding your affinity tag, the histidine. Any steps to or need to later remove it when you want to do some in vivo challenges to verify for the immunity of your produced protein?
Yeah, so basically I will start by saying that there's already some publication that showed that this histidine by itself can be immunogenic. So yes, it would be very important to remove it before doing immunogenicity assays in vivo. And for that, normally in the plasmid before the histidine tag, there's a cleavage site. So once you have purified your protein, you can do a digestion and then just remove this tag and have your protein in its native structure.
Okay. Any other words of the wise before we move to the other presentation? And if ever the attendees have some questions, please do not hesitate to write your question in the Q&A box at the bottom of the screen. So any other words of wisdom?
Don't desperate and keep going, and everything will be fine. Pretty sure.
Yes, patience and resilience. I think these are the key words in protein production. Thank you very much, Nicolas, for your presentation.